To understand Mendel's idea and question about dihybrid inheritance, we first need to understand the concept known as independent events and the concept known as linked events. So imagine there are two coins. Okay? One coin is either head or tail on one side, while for another coin, it's either gold or silver on one side. If I flip these two coins, I would like to know the probability of two events happening at the same time. Say, for example, I want to know what is the probability of having head and having gold at the same time. Well, this answer depends on whether what the relationship between these two events. So event 1 and event 2. I think most of you would at once tell me that the probability of having a head and gold would be 1/4, because you know that the probability should be equal to 1/2, which is having a head, times 1/2, which is probability of having gold, and eventually it leads to 1/4. I'm not saying that you are correct or wrong, but I'm saying that when you think like this, you actually assumed one thing. You assumed that the flipping of this coin would not affect the flipping of this coin. In other words, you are saying that the event here is independent or will not affect the other event. In this way, we say the two events are independent, meaning that these two events won't affect each other. But imagine there is actually another possibility. Imagine that these two coins are now connected by a rod. So that now, whenever you flip this whole thing, if the head comes up because of the rod, then it must be the gold side that comes up. Whenever the tail side comes up, the silver side comes up. They flip as a single unit. Now we say that these two events, they are no longer independent, because one event would affect the outcome of the other event. We say these two events are linked. In this way, the probability of having head and gold at the same time is no longer 1/4, but is actually 1/2 by counting, because it can only either be head and gold or tail and silver. I hope this case illustrates to you the meaning of the word independent and the meaning of the word linked. It means that there are two possible relationships between two events. Let's take this concept to another level. Imagine I now don't show you the coins. I flip these coins in a black box, let's say. And you don't know whether these two coins are linked or not. So how can I do an experiment to check whether these two coins are linked or not linked? So pause the video for a while and think about it. Well, the answer is, imagine you can't see the coin, but you can flip it and you can check the outcome. You can flip it by a lot of times and really check the probability of having head and gold. If the probability of having head and gold is 1/4, then you know that the two coins are not linked. They are independent of each other. But if the two coins, if you flip them, the probability is really 1/2 of having head and gold, then you can deduce that these two coins should be linked. This is the experimental idea for checking whether two events are independent or linked. Now, why is this concept linked or related to inheritance? Because when living things inherit their characteristics, we actually inherit a lot of different characteristics at the same time. Each characteristic can be seen as a single event. The inheritance of one character is a single event. The inheritance of another character is another event. So then Mendel thinks about this. Alright, now when I do work on the pea plant, I have a good understanding about how a single character is inherited. But now, what if I consider the inheritance of two characters at the same time? When two events or when two characters are inherited at the same time, like the coins here, there are two possible relationships between these two inheritance. They can be independent or they can be linked. In other words, meaning that they can affect each other. So Mendel's main question would be, will the inheritance of one character affect, or is it linked to the inheritance of another character? This is the big concept that he is trying to test. So let's apply this into an example. Imagine I consider two characteristics, seed color and seed shape. Imagine that I'm trying to study the inheritance of seed color and seed shape together, and there is a plant that is heterozygous for both, so that it is yellow and round seed. So when it self-fertilizes, what will be the outcome and the probability of the different outcomes. So I can use letter A to represent the genes or hereditary factor at that time. For seed color, I can use letter B, a different letter, to represent seed shape, another character that is. So different letters for different character. Since it is heterozygous for both seed color and seed shape, it means that now I'm considering two events, big A small a crossed with big A small a, because heterozygous for seed color, and at the same time this is big B small b crossed with big B small b for seed shape. By what we have already learned, we know the the probability of each outcome. So that means if I consider seed color alone, what is the probability of having yellow or green seed? Pause and think for a while. The answer is, just go back to this traditional cross. And you'll remember that 3/4 is the probability of having yellow seeds, and the probability of having green seeds is 1/4. The same can be applied to seed shape. The probability of having round seeds would be 3/4, and the probability of having wrinkled seeds would be 1/4. If I consider the inheritance of each character alone, that would be the concept of monohybrid inheritance. But the key is that now Mendel is trying to study the inheritance of these two factors together. So what will happen when they inherit together? Will they affect each other? That, in other words, he is asking the question whether the inheritance of seed color is independent or linked to the inheritance of seed shape. How can Mendel, or how can we solve this problem experimentally? Because we cannot see how the hereditary factors work. Just like the case we have discussed, let's say we cannot see the coins physically. In Mendel's case, he cannot see how the gene or the hereditary factor is working. So he got to use experimental results to deduce the relationship between these two events. So how can he do that? You can pause this video for a while and think about it. If you want some hints, go back to the part of the video when we are discussing how we can know whether these two coins are linked or not. To experimentally solve the problem, that means to find the relationship between the two inheritance, we first need to predict the probability of different outcomes. Say, for example, if we assume the two events to be independent, we can actually calculate the probability of different outcomes. The probability of having yellow and round seed would be 3/4, that is the probability of having yellow, times the probability of being round seed, that is 3/4 as well. That gives you 9/16. We can do the same for different outcomes by multiplying the probability of the single events and find out the probability of each event. Then, just like the coin story, we really do the breeding. We really cross a heterozygous plant with yellow and round seeds. We let it self-fertilize to produce a large amount of offspring plants, and then study their seeds. And by counting the seeds, we can trace or study the actual probability of these events and try to match those actual numbers with the predicted values here. If the actual probability matches these calculated values, then it shows that these two events are independent. If not, if the probability of these events greatly differ from these numbers, then it shows that the two events are linked. What I show here is the actual results of Mendel's cross. What can you conclude from these actual results? If you really calculate the ratios, you would see that these numbers match with these probabilities very well, indicating that the inheritance of seed color is independent to the inheritance of seed shape. Mendel's next question would be, why would they be independent? How can we explain what happens? Mendel tried to use the same mechanism as he used in monohybrid inheritance to explain what happens during dihybrid inheritance. Mainly, this diagram is trying to illustrate why the inheritance of seed color can be independent to the inheritance of seed shape. So referring to the story in the previous part of the video, a plant with yellow and round seed self-fertilize. And that parent plant is actually a hybrid. So we illustrated the genotype of the parents here. Remember Mendel's first law? Mendel says that during gamete formation, each allele for a gene would separate, and each gamete would receive only one allele. So if this is in the old days, is the genotype is big A small a, then the gamete would either get a single big A or single small a. Well, this principle, this first law, is still applied here, meaning that the gametes would, the A gene, that means the gene for seed color, would separate, and the gametes would only get one allele for seed color. But that's not the end for gamete formation. Don't forget that now we also consider the genes for seed shape. So this pair of gene for seed shape would also separate so that the gamete would receive one allele for seed shape, meaning that there should be a letter B here. So first of all, it means that the gametes would have two letters here, or in other words, two genes. A gene for seed color and a gene for seed shape. This is applying Mendel's first law. Remember, this idea is still the same as a monohybrid inheritance. What is something very new is that we have to explain why the two events are independent. So Mendel then developed a second law of inheritance. He suggests that when the two genes, when the alleles separate, the separation of one pair of allele does not affect, or in other words, independent to the separation of another pair. What does it mean? And how does it affect gamete formation? It now means that, with this second law in mind, it means that if the separation of the first pair gives the gamete big A, this separation does not affect or does not determine the outcome in the separation of the other pair. Meaning that this can either be big B or small b. Or I can draw it separately, like this. That is the key concept in explaining why the two events are independent. The separation of one pair of allele is independent to the separation of another pair of allele, meaning that this big A can be combined with big B or small b in the gametes. But don't forget, the outcome of the first separation of the seed color gene, that means letter A, it can be small a, right? So now it means there can be other two possibilities for gametes. It can be A, small a together with big B, or small a together with small b. let's review this gamete formation process again and see how this illustrates Mendel's idea. Mendel thinks that the first law still stands. That means each pair of allele would separate so that the gamete would get one member for each allele, for each pair of allele. So that means the gamete must have a letter A and must have a letter B. To explain the independent nature of the two events, he proposed that the separation of one pair of allele is independent to the separation of another, so that when a gamete gets a big A, it can actually get either big B or small b. This idea is now known as Mendel's second law of inheritance. By applying these two laws together, this parent now can produce 4 possible types of gametes. Now, of course, the same can be applied for the other parent. So that means each parent now produces 4 types of gametes. The next stage of inheritance is randomly pick one of the gametes to fertilize with another gamete to see or to give an offspring with a particular genotype. Say, for example, if this gamete and this gamete fertilize, then the genotype of the F1 would be ABB like this. Now, of course, you can repeat this for every possible combinations, but the diagram would be very chaotic. So if you are trying to fertilize or find all the outcomes by fertilizing a lot of different possible gametes, then you can use a tool called Punnett square, which allows you to study the fertilization outcome in an easy way. To set up a Punnett square, we list all the gametes from one parent on one side. Then we list the gametes of another parent on the other side. Now, in this particular case, since each parent gives 4 possible gametes, therefore we have a 4x4 table creating 16 possible outcomes. Each box here represents an outcome of fertilization. Say, for example, this box is the fertilization outcome of big A small b and big A big B. So therefore, the genotype of the offspring here would be like this. And we can also determine the phenotype of this offspring. Alright, then pause this video and take some time to, complete this table. So after completing the Punnett square, we now have to study the phenotypic ratio of the F1. So we need to first list out all possible phenotypes of F1. My way of thinking is something like this. I first think about dominant for both case, that is yellow and round seed. Then I consider having either one phenotype as the dominant one. Say for example, a dominant seed color combining with a recessive seed shape, or a dominant seed shape but combining with the recessive seed color. So that means these middle two is having one dominant phenotype with one recessive phenotype. And at last, the one with both phenotypes to be recessive. So in this way of thinking, you won't miss any possible outcome. So then we need to find their ratios by counting the outcomes in the Punnett square. Now at first sight, it's quite difficult to read, right? So what I suggest you to do, and I will do this in my own work, is that I take our highlighter to highlight all the dominant phenotypes. Say for example, yellow is the dominant one, so I highlight the ones that are having yellow color. Alright, like this. Then I'll do the same for seed shape. I highlight the dominant phenotype, like this. So now it's easier, much easier for me to count. When I want to look for yellow round seeds in F1, it means that both boxes are highlighted. So therefore, by counting 1, 2, 3, 4, 5, 6, 7, 8, 9. So there are 9 boxes, both highlighted. When I look for yellow and wrinkled seeds, it means that I want to find only the first box is highlighted. So 1, 2, and 3 here. Green and round, then I look for only the second box phenotype is highlighted. So 1, 2, and 3 here. Finally, the green wrinkled one is this one with no highlight, so 1 there. So the phenotypic ratio would be 9 to 3 to 3 to 1, and this is quite typical for a double heterozygous parent to fertilize. Now let's spend a little time here to appreciate this ratio and how it matches with the idea of independent events. Say for example, now I want to see what is the probability of having yellow and round seed. So the probability equals to 9 over 9 plus 3 plus 3 plus 1. That's 16. Why is this number so amazing? If we consider yellow or the seed color and seed shape independently, having the probability of having yellow seeds alone is actually 3 over 4. The probability of having round seeds here again is actually 3 over 4. So this number 9 over 16 is so amazing because it's exactly the same as by mathematical calculation, we multiply the two probabilities together. We only multiply probabilities when the two events are independent. So this number is amazing because it matches with the prediction when the two events are independent. You can check this for all other possible outcomes. Now, another amazing thing about this probability is that it doesn't differ or violate Mendel's monohybrid inheritance. So imagine now, I ignore the seed shape, and only consider the probability of the seed color to be yellow. In the old days, when we don't know about dihybrid inheritance, we will simply do this, right? We don't know there is seed shape there. We ignore that. So that means we ignore the gene for seed shape. We ignore letter B. In this way, the probability outcome would be 3/4. Just like we do in the old days, right? This is the answer we have when we consider monohybrid inheritance. Does it apply here, or is the result the same here? To find out the probability of having yellow seeds, that means we have to count all the possible outcomes that are yellow. So I can check the boxes to be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Or I can do this by adding these two numbers up, because both of them have yellow seeds. So that means the probability would be 9 plus 3, that means 12, over the total possible outcome, 16. You see? Same answer as the case we do in the monohybrid inheritance. Why is the answer the same? Because the two events are independent of each other, and the Mendel's first law is still applied when the gametes are formed. So that's why the outcome is the same. I hope this video gives you an idea how Mendel explained dihybrid inheritance, and it also shows you how in tests or exams we can solve dihybrid inheritance problems using a formal genetic diagrams like this. So from the first and second part of the video, we can see that Mendel's discovered that the inheritance of seed color and seed shape are independent to each other. But why? Now, Mendel's explanation, based on his proposed Mendel's second law, is that the separation of alleles for a gene is independent to the separation of the alleles of another gene. Meaning that if the gametes get a big A here, the same gamete may get big B or small b. Well, that mathematical model fits the outcome. But is this true? Is this what really happening in our body at our cell level during gamete formation? So in this part of the video, we try to look for the modern explanation, we try to look at the events at the cell and chromosome level to explain or to support Mendel's second law, why the alleles of different genes separate independently. Well, we are going to look at the gamete formation of a single parent, big A small a, with a genotype big A small a, big B small b. I'm looking at a single individual, okay? A single parent. More at chromosome level, we now know that the gene for seed color is located in a different homologous pair than the gene controlling the seed shape. These two genes are located on different homologous pairs. We can explain, or we can illustrate by a drawing like this. Now this is the cell that is going to form gametes. It's not yet a gamete, but it's going to form a gamete by meiosis. Remember that in meiosis, homologous chromosomes pair up like this. And then the homologous pairs line up in the middle of the cell like this. Then the chromosomes of each homologous pair would separate. So these chromosomes are being pulled away and they are being separated like this. So what will be the contents of the chromosome in the gametes? So pause and think for a while. What will be the content of the chromosomes in the gametes? So the gametes would look like this. Well, this explains why the parent would produce gametes big A, big B, and big A, small b. Oh, sorry, and small a, small b. But if you remember what we have taught, there should be 4 possible gametes, right? So how come the two events, the inheritance of the two events are independent? Well, it is because if you remember what we learned in meiosis, there is a process known as, there is a stage known as independent assortment. Meaning that when these pairs line up in the middle of the cell, the lining up of these two are independent and they don't affect each other, meaning that there is another possibility how these pairs are arranged and lined up in the middle. Can you imagine how the other possible arrangement would look like? The other possible arrangement would be swapping the position of these two chromosomes so that the small b is lined up here and the chromosome with the big B is lined up here. Now if the lineup is different, then when the chromosomes are being separated during anaphase 1, how would the outcome differ? So you can see it gives two types of gametes, big A small b, small a big B, which does not exist here. Now remember that both alignment is possible. So when you make a lot of gametes, it actually produces all 4 types of gametes. Big A, small b, big small a, big B as the other possible arrangement. And together, these 4 are all possible. These 4 are all possible. So, this is the explanation of Mendel's second law at the chromosome level. Why are the separations of the alleles of a gene independent from another? It is because 1, the genes are located on different chromosomes. And the second point is that during meiosis, these chromosomes would be assorted independently, meaning that the separation of the chromosomes of a homologous pair is independent to the separation of another. So therefore, there are 2 possible lineups here, creating a total of 4 possible types of gametes here. So this explains how each parent works. And of course, this applies to every parent. So that's why eventually the inheritance of seed color is independent to the inheritance of seed shape.